Hello dear students, welcome back once again to Kamala Online Learning. We are back again with another informative video lesson from your schedule. Please be completely attentive during the teaching. Keep your textbook and also a rough book with you. Note down important points and difficult or new words as we go. This video lesson will begin with an overview which will give you an idea about the contents of the lesson. It will be followed by learning objectives. Please note down the learning objectives in your notes and try to answer them at the end of the session. If you have any doubts, rewatch the video and refer to your textbook and study material. In case you need further clarification, please mail your doubts to kamalaonlinelearning@gmail.com. This video is recorded with simple, easy to understand language and eye-catching graphics. We hope it helps you to understand the concepts better. So, let's get started.
Welcome to the chapter Cell, the basic unit of life.
This slide presents the overview of the chapter. Learning objectives. By the end of this chapter, you will be able to
Understand the importance of the cell. Explain the discovery of the cell. Identify the nucleus in plant cell and animal cell. Explain the discovery of the nucleus. Interpret the diversity in cells. Identify different groups in the transverse section of dicot stem. Identify different parts of nerve cell, striated muscles and unstriated muscles. Know the size of different organisms.
Introduction. Before entering into the chapter, follow the instructions shown on the screen. Click each tab to know.
In lower classes, we have learned that our earth is a beautiful place wherein different types of living organisms live happily. All living beings are composed of cells, from tiny masses to huge conifers and from invisible bacteria to huge blue whales. All have a basic unit called cell. All living organisms carry out certain functions. Do you know what is the basic structural unit of organ? Who discovered the cell? Diversity in cells. What is the nucleus? Let us try to answer these questions and learn more about the cell and its importance. Cell. Cells are very small in size, which cannot be seen through the naked eye. To observe these minute cells, one needs a microscope. Around 350 years ago, before the invention of microscope, people were not aware of the living world that was not visible to the naked eye. Thereafter, with the help of the microscope, many scientists like Athanasius Kircher Jan Swammerdam, Antony van Leeuwenhoek and Robert Hooke started observing and describing the unknown world. Among these scientists, Antony van Leeuwenhoek was the first human to see living bodies like bacteria, yeast, protozoa, red blood cells, and the teeming life in a drop of water. He prepared several types of magnifying glasses and used his lenses to study about different types of living and non-living things. His microscope consisted of a single biconvex lens and was termed as the simple microscope. To study about basic structures, it is important to know the proper usage of microscope and the preparation of microscopic slides. Now, let us study about the discovery of the cell. Discovery of the cell. In the year 1665, a British scientist named Robert Hooke discovered a cell. He developed a compound microscope by using two lenses in order to achieve greater magnification. He studied a thin piece of cork, soft bark from oak tree, under his microscope. From the microscope, he observed that the cork piece looked similar to the structure of honeycomb with many empty spaces or empty box-like structures. He thought that these empty spaces are made up of very small cavities. He called these cavities as cell. This was for the first time to be observed that living things consist of separate units called cell. In Latin, the word cell refers to little room. Now, let us see what Robert Hooke might have observed in the cork.
Knowledge check. Attempt the following questions to check your understanding.
Here, let us perform an activity to observe the cells in matchstick. Take a matchstick and soak it in water for half an hour. Next, with the help of a blade or a knife, cut the matchstick into thin pieces. Choose a thin piece and place it on a slide. Next, put a drop of water on the slide and cover it with a cover slip. Now place the slide under a microscope and observe it. It is observed that the cells of the matchstick under a microscope are similar to the cells of the cork of an oak tree, as we have observed previously. From this experiment, we have learned that the cells of a matchstick and the cork of an oak tree are similar. This is because both the cells of cork and matchstick are dead cells. The discovery of cell by Robert Hooke was a milestone in the history of science. Take a piece of onion and cut out a small fleshy portion from the bulb. Break this fleshy piece into two halves and you will find a thin transparent peel holding the piece together. Now, take out the peel and cut a small piece from it. Next, take a permanent slide and place a peel on it. Next, add a drop of water on the slide and spread the peel evenly. Cover the slide with a cover slip. Next, place the permanent slide mount under a microscope and observe it. Draw the image of what you have observed along with labels. It is observed that all the cells are similar. The cells are in rectangular or linear in shape and all the cells are firmly bounded together. From the experiment, we learned that all the cells of onion are similar in shape irrespective of the size. The onion peel cells are the plant cells. Now, let us do an activity to observe human cheek cells. Precautions. Before starting the activity, some precautions ought to be followed. Wash the spoon thoroughly before using it. Do not scrape the cheek too hard because it may injure you. Scraped material should be spread uniformly on the slide. See that there should not be any air bubbles under the cover slip. Excess stains should be drained off on the permanent slide. Now, let us do the activity. Take a permanent slide and add a drop of water to it. Next, take a wooden spoon or a toothpick. With the help of the toothpick, scrape the inner surface of the cheek. Note, scrape the cheek with the flat end side of the toothpick. Now, remove the toothpick from the mouth and place it in a drop of water taken on slide. Next, spread the toothpick in a drop of water to enable our cheek cells to spread out. Now, remove the toothpick from the slide and cover it with a cover slip. Now place the slide under a microscope and observe it. It is observed that the shape of the cheek cells are irregular. A dark-colored border is observed for each cheek cell. From the experiment, we learned that the cells of cheek cells are irregular in shape. A scientist named Robert Brown made great contributions towards cells. Before Robert Brown, it is believed that another scientist named Felice Fontana have observed the nucleus in the epithelial cells, which is the outermost layer of animal body, in the 18th century. But the credit for observing the nucleus in different kinds of cells and finding that it was an integral part of all cells goes to Robert Brown. While observing the cells in orchard leaves, Brown observed a small near-circular spot which was slightly more opaque than the surrounding parts. He stated that this structure was present inside the cell and he named it as nucleus. Among the different parts of the cell, the nucleus is the most important and well-known part. He also observed this nucleus in all the other cells. He found this nucleus in the year 1831. There was a gap of around 180 years between the year of first observing the cells, around 1650, to the year of observing the nucleus, 1831. The image shown on the screen was the plant cell which was observed by Robert Brown when he observed the nucleus for the first time. 3 stomata can also be clearly seen in the image. These stomata are the breathing pores through which a plant exchanges gases with the atmosphere. Here, let us perform an activity to observe the nucleus in onion peel. Take a piece of onion and cut out a small fleshy portion from the bulb. Break this fleshy piece into 2 halves and you will find a thin transparent peel holding the piece together. Now, take out the peel and cut a small piece from it. Next, take a permanent slide and place a peel on it. Now, add 1 or 2 drops of safranin and methylene blue to the slide. Next, cover the slide with a cover slip and leave it aside for 5 minutes. Next, add water dropwise from one side of cover slip and remove the extra water with the help of filter paper from the other side. Now, place the permanent slide mount under microscope and observe it. Blue-colored spots are observed in each cell. A thick border is observed for each cell. In between the dark-colored spot, and the border of the cell, a jelly-like substance is observed. The blue-colored spot observed within the cell is called nucleus. The dark outer boundary of a cell is called cell membrane. The jelly-like substance present between the nucleus and cell membrane is called cytoplasm. Now, let us observe the nucleus in our cheek cells and find the difference between the cells of an onion peel and the cheek cells. Take a permanent slide and add a few drops of methylene blue to it. Next, take a toothpick and with its help, scrape the inner surface of the cheek. Note, scrape the cheek with the flat end side of a toothpick. Remove the toothpick from the mouth and place it on the dye and spread the toothpick in the dye to enable our cheek cells to spread out. Remove the toothpick from the dye and cover it with a cover slip. Next, place the permanent slide mount under the microscope and observe it. A dark blue-colored spot is observed at the center of each cell. An outer boundary is observed for each cell. In between the dark-colored spot and the border of the cell, a jelly-like substance is observed. The dark blue-colored spot observed within the cell is called nucleus. The outer boundary of a cell is called cell membrane. The jelly-like substance present between the nucleus and the cell membrane is called cytoplasm. The cell membrane gives a definite shape to the cell. It selectively allows the substances to pass in or out of the cell. When compared to cheek cells, the outer covering of onion peel cells is much clearer. This is because another layer called cell wall is present over the cell membrane. The cell wall provides rigidity to the cell. The nucleus is present in both the onion peel cells and the cheek cells. But in case of onion peel cells, the nucleus is not in the center, whereas in case of the cheek cells, the nucleus is present in and around the center. The cytoplasm is a very heterogeneous material. Cytoplasm contains of membrane-bounded nucleus called cell organelles and complex chemicals. The cell organelles help in carrying out several functions within the cell.